Welcome to this postgraduate lecture on the histological structure and functions of the adrenal and pineal glands. This video will cover their microscopic organization, hormone synthesis, physiological roles, and clinical importance. The endocrine system is a complex network of glands that coordinate body functions through chemical messengers, known as hormones. Unlike exocrine glands, endocrine glands do not possess ducts. Instead, their secretions enter the blood and travel to target organs. Major endocrine organs include the hypothalamus, pituitary gland, thyroid gland, parathyroid glands, adrenal glands, pancreas, gonads, and pineal gland. These glands regulate growth, metabolism, reproduction, stress response, water and electrolyte balance, and circadian rhythms, thereby maintaining internal homeostasis. The adrenal glands are paired endocrine organs located on the superior poles of the kidneys and are therefore also known as suprarenal glands. Each adrenal gland consists of two functionally and embryologically distinct regions, the outer adrenal cortex and the inner adrenal medulla. The adrenal cortex produces steroid hormones involved in metabolism, electrolyte balance, and sexual development, while the medulla produces catecholamines responsible for the immediate stress response. The pineal gland, on the other hand, is a small neuroendocrine structure located within the epithalamus of the brain. It secretes melatonin, a hormone that regulates biological rhythms and the sleep-wake cycle. By the end of this lecture, students should be able to describe the anatomical location and embryological origin of the adrenal and pineal glands. Students should also be able to identify the histological layers, recognize the different cell types under the microscope, explain the hormones secreted by each gland, and understand their physiological functions. Finally, we will discuss important clinical disorders associated with abnormal hormonal secretion, which will provide a correlation between histological structure and disease. The adrenal glands are small, yellowish, highly vascular endocrine organs, situated retroperitoneally above each kidney. The right adrenal gland typically has a pyramidal shape, whereas the left adrenal gland is semilunar or crescent-shaped. Each gland weighs approximately 4 to 6 grams and is surrounded by a connective tissue capsule. Despite their small size, the adrenal glands are essential for survival because they regulate metabolism, blood pressure, electrolyte balance, and the body's response to physical and emotional stress. The adrenal gland has a unique dual embryological origin. The adrenal cortex develops from mesoderm, specifically from the coelomic epithelium, whereas the adrenal medulla originates from neural crest cells. During development, neural crest cells migrate into the developing cortex and differentiate into chromaffin cells. This difference in embryological origin explains why the cortex functions as a steroid-producing endocrine tissue, while the medulla acts as a modified sympathetic nervous structure. The adrenal gland is enclosed by a capsule providing structural support. It is divided into two main regions, the outer cortex, making up about 90% of the volume, and the highly vascularized inner core, the medulla, accounting for the remaining 10%. The adrenal cortex is subdivided into 3 concentric zones based on cell arrangement. From the capsule inward, we find the zona glomerulosa, fasciculata, and reticularis. The zona glomerulosa is the outermost layer, featuring cells in arched cords, and is the site of mineralocorticoid synthesis, producing aldosterone. Aldosterone maintains blood pressure by promoting sodium and water reabsorption in the kidneys. Aldosterone secretion is regulated by the RAAS pathway. A drop in blood pressure prompts renin release, leading to the formation of angiotensin II, which stimulates aldosterone secretion. Clinical conditions can cause imbalances, such as hypertension or hypotension. The zona fasciculata constitutes nearly 75 to 80% of the total adrenal cortex. Histologically, it is composed of large polyhedral cells known as spongiocytes. These cells are arranged in long, straight cords that extend radially from the zona glomerulosa toward the adrenal medulla. The cells appear pale and vacuolated under the microscope because they contain abundant lipid droplets that are removed during tissue processing. These lipid droplets contain cholesterol, which serves as the precursor for steroid hormone synthesis. The cytoplasm of these cells is rich in smooth endoplasmic reticulum and mitochondria with tubular cristae, characteristic features of steroid-secreting cells. Between the cords of spongiocytes, numerous fenestrated sinusoidal capillaries are P. Cortisol is the principal glucocorticoid hormone of the human body and affects nearly every tissue. Its major metabolic function is to increase blood glucose levels by stimulating gluconeogenesis in the liver. It promotes the breakdown of proteins into amino acids and enhances lipolysis, providing substrates for energy production during periods of stress. Cortisol also possesses powerful anti-inflammatory and immunosuppressive effects. It inhibits inflammatory mediators and reduces the activity of immune cells. During physical or emotional stress, Increased cortisol secretion helps the body maintain energy balance and adapt to challenging conditions. However, abnormal cortisol levels can lead to serious disorders. Excess cortisol results in Cushing syndrome, whereas cortisol deficiency, the secretion of cortisol, is controlled by the hypothalamic pituitary adrenal axis, commonly called the HPA axis. The process begins in the hypothalamus, which releases corticotropin-releasing hormone, or CRH. CRH stimulates the anterior pituitary gland to secrete adrenocorticotropic hormone, known as ACTH. ACTH travels through the bloodstream and stimulates the zona fasciculata of the adrenal cortex to synthesize and release cortisol. Once cortisol levels rise in the blood, it exerts a negative feedback effect by inhibiting further secretion of CRH from the hypothalamus and ACTH from the pituitary gland. This feedback mechanism maintains cortisol concentration within the normal physiological range. The innermost layer of the adrenal cortex is the zona reticularis, which lies directly adjacent to the adrenal medulla. Under the microscope, the cells of this layer are smaller and darker compared with those of the zona fasciculata. They are arranged in irregular interconnected cords forming a network-like pattern, which gives this region the name reticularis. The cytoplasm contains fewer lipid droplets and often contains lipofuscin pigment, producing a brownish coloration in some cells. The zona reticularis synthesizes adrenal androgens, particularly dehydroepiandrosterone, commonly known as DHEA, and androstenedione. Adrenal androgens are weaker sex hormones that serve as precursors for the synthesis of testosterone and estrogens in peripheral tissues. These hormones contribute to the development of secondary sexual characteristics, especially the growth of pubic and axillary hair. In females, adrenal androgens are an important source of androgenic hormones and influence libido. The secretion of adrenal androgens is mainly regulated by ACTH from the anterior pituitary. Excess androgen production can cause virilization in females and precocious puberty in children, whereas deficiency may lead to reduced growth of pubic and axillary hair. Before proceeding to the adrenal medulla, let us compare the three cortical zones. The outer zona glomerulosa contains cells arranged in rounded clusters and produces mineralocorticoids, primarily aldosterone, which regulate salt and water balance. The middle zona fasciculata contains straight cords of lipid-rich spongiocytes and produces glucocorticoids, mainly cortisol, responsible for metabolism and stress response. The inner zona reticularis contains a network of smaller cells that produce adrenal androgens involved in sexual development. A useful mnemonic for remembering these zones is GFR, salt, sugar, sex, where the zona glomerulosa controls salt balance, the zona fasciculata regulates sugar metabolism, and the zona reticularis influences sex hormone production. The adrenal medulla forms the central region of the adrenal gland and functions as a modified sympathetic ganglion. Unlike the cortex, which originates from mesoderm, the adrenal medulla develops from neural crest cells. This common origin with the sympathetic nervous system explains its role in rapid stress responses. The medulla consists primarily of chromaffin cells, which are modified postganglionic sympathetic neurons. These cells are closely associated with a rich network of blood sinusoids and receive direct stimulation from preganglionic sympathetic nerve fibers. The hormones produced by the adrenal medulla are catecholamines, which prepare the body for emergency situations. Chromaffin cells are the characteristic endocrine cells of the adrenal medulla. These cells are large, polygonal, and arranged in clusters or cords surrounding sinusoidal capillaries. Their cytoplasm contains numerous secretory granules filled with catecholamines. The term chromaffin originates from the brown coloration produced when these cells react with chromium salts, known as the chromaffin reaction. There are two major populations of chromaffin cells. Approximately 80% produce epinephrine, while the remaining 20% produce norepinephrine. Although these cells are derived from neurons, they lack axons and dendrites and release their secretions directly into the bloodstream. The synthesis pathway starts with tyrosine, which is hydroxylated to L-DOPA. L-DOPA is decarboxylated to form dopamine, which is then hydroxylated to norepinephrine. Norepinephrine is finally methylated to produce epinephrine. Catecholamines elicit widespread systemic effects. These include cardiovascular effects like increased heart rate and blood pressure, respiratory bronchodilation, metabolic effects like glycogen breakdown, and neurological effects such as heightened alertness. These collective effects optimize the organism for survival. The adrenal glands receive a robust arterial supply from 3 suprarenal arteries. Microcirculation involves centripetal flow through the cortex, delivering steroid-rich blood to the medulla. Venous drainage converges into a single central medullary vein. The adrenal gland features dual regulatory systems. The cortex is hormonally regulated by the RAAS and ACTH, leading to a slower and sustained response. The medulla is neurally regulated by sympathetic stimulation, resulting in a rapid, immediate response. The adrenal medulla is considered a modified sympathetic ganglion due to its neural crest embryological origin and innervation by preganglionic sympathetic fibers. Chromaffin cells function as specialized postganglionic neurons that secrete hormones directly into the blood. Microscopic identification of the adrenal gland starts with the capsule. Moving inward, we find the zona glomerulosa in small clusters, the zona fasciculata in thick pale cords, the zona reticularis in a dark irregular network, and the basophilic adrenal medulla. Addison's disease is primary adrenocortical insufficiency caused by the destruction of the adrenal cortex, which leads to deficiencies in both cortisol and aldosterone. A characteristic clinical sign is skin hyperpigmentation due to increased ACTH production. Cushing's syndrome, or hypercortisolism, results from chronic exposure to excess glucocorticoids. Clinical features include moon phases, central obesity, buffalo hump, and thin limbs from muscle wasting. Other signs are violaceous abdominal striae, easy bruising, and secondary conditions like hyperglycemia and hypertension. Conn syndrome, or primary hyperaldosteronism, features resistant hypertension, sodium retention, hypokalemia, and muscle weakness. Congenital adrenal hyperplasia, an autosomal recessive condition, is caused by 21-hydroxylase deficiency. This enzymatic block shunts precursors toward androgen production, leading to virilization and precocious puberty, alongside adrenal enlargement. Pheochromocytoma is a catecholamine-secreting neuroendocrine neoplasm. The classic triad includes severe episodic headache, profuse diaphoresis, and tachycardia. Hypertension and anxiety are also prominent. The diagnostic key is elevated plasma-free metanephrines. Imaging reveals the adrenal mass, and other tumors include benign adenomas and aggressive cortical carcinomas. The adrenal cortex has 3 functional zones. The zona glomerulosa produces mineralocorticoids like aldosterone, maintaining salt and water balance. The zona fasciculata produces glucocorticoids like cortisol, regulating glucose metabolism and stress response. The zona reticularis produces androgens like DHEA. The medulla secretes catecholamines like epinephrine for the fight-or-flight response. The mnemonic GFR, salt, sugar, sex, helps remember this zonation. We now transition from the adrenal gland's rapid, survival-oriented stress management to the pineal gland's subtle, rhythm-based chronobiological modulation. The pineal gland is the primary pacemaker, translating light information into chemical signals via melatonin secretion to govern circadian rhythms. The pineal gland is a small neuroendocrine organ, sometimes called the third eye, and lacks a typical blood-brain barrier. It's strategically located in the hypothalamus, attached to the posterior wall of the third ventricle. Its primary function is the synthesis of melatonin, critical for circadian regulation. The pineal gland originates from a midline evagination of neuroectoderm in the diencephalon roof. It matures from a pineal diverticulum into a conical structure by approximately 8 weeks. Anatomically, it's attached to the posterior third ventricle by a short stalk. It is covered by a pia mater capsule and has a rich arterial supply. The pineal gland parenchyma is organized into lobules by septa and capillaries. The main cell type is the pinealocyte, responsible for secretion. Scattered interstitial glial cells, similar to CNS astrocytes, provide support. Corpora arenacea, or pineal sand, are calcified concretions unique to the pineal gland, serving as radiological markers. Pinealocytes are the principal secretory cells. They are large, irregular cells with active nuclei and long-branching cytoplasmic processes that terminate near fenestrated capillaries. They are rich in organelles, including a prominent Golgi apparatus, and contain secretory granules. Melatonin synthesis is a key function, modulated by light signals relayed from the retina. Interstitial glial cells are the pineal gland's supporting microenvironment. They have smaller, darker nuclei and resemble CNS astrocytes. They are located along connective tissue septa for vascular support, providing structural scaffolding, and maintaining the metabolic environment for pinealocytes. They stain positive for glial fibrillary acidic protein, confirming their glial lineage. Pineal sand, or corpora arenacea, are basophilic, concentric, and lamellated calcium deposits. This is a normal physiological process, with concretions increasing in size and number with age. Radiologically, pineal sand is a critical midline landmark on non-contrast CT, allowing detection of mass effects, midline shifts, or lesions. Melatonin synthesis is a multi-step biochemical pathway starting from tryptophan. Photic signals from the retina travel to the SCN, which relays information to the pineal gland. Darkness stimulates the sympathetic pathway, upregulating the rate-limiting enzyme AANAT and increasing melatonin production, while light rapidly suppresses it. Melatonin is the master synchronizer of circadian rhythms, primarily signaling darkness and regulating the sleep-wake cycle. It also impacts the reproductive system by modulating GnRH release. Additionally, melatonin provides cellular protection as a potent endogenous antioxidant, protecting against oxidative stress and modulating the immune system and body temperature. The pineal gland's clinical significance involves structural pathologies and circadian disruptions. Tumor types include pineocytomas, pineoblastomas, and germ cell tumors. Mass lesions can lead to Paranose syndrome, causing upward gaze palsy and hydrocephalus, obstructing the aqueduct of Sylvius. Loss of melatonin inhibition can cause precocious puberty. Circadian disorders include chronic insomnia and jet lag. This video has explored the anatomy, histology, and clinical pathology of the adrenal and pineal glands, covering their unique structures and vital roles in systemic health and chronobiology. The adrenal and pineal glands differ fundamentally in location, embryology, histology, and function. The adrenal gland is paired and retroperitoneal, managing stress and metabolism, while the single pineal gland is central in the brain, strictly regulating chronobiology and circadian rhythms via melatonin. Adrenal hormones handle blood pressure, metabolism, and the acute stress response. The pineal gland's melatonin acts as a neuroendocrine transducer, synchronizing systemic biology with environmental light-dark cycles, making it key to chronobiology. Adrenal regulation involves the hypothalamic pituitary adrenal, or HPA axis for classic negative feedback, and the renin-angiotensin-aldosterone system, or RAS, using hemodynamic sensors. Pineal regulation is unique, employing a neuroendocrine circuit based on environmental light exposure. Adrenal disorders include Addison's disease, Cushing's syndrome, Conn syndrome, and pheochromocytoma. Pineal pathologies involve mass lesions leading to Paranose syndrome and functional disorders related to melatonin and sleep disruption. The visual summary highlights the distinct hallmark clinical findings. In summary, the adrenal cortex has the GFR zonal architecture, glomerulosa, fasciculata, and reticularis. For the pineal gland, locating corpora arenacea, or pineal sand, is essential for definitive histological confirmation and as a radiological marker.